Assalamualaikum and a very good morning to examiners. My name is Siti Nuriani binti Jaafar. I am final year student and I would like to present to you about my study. The title of my study is Morphometric Analysis of Genus Cyclocheilichthys Cyprinidae in Peninsular Malaysia. Cyprinidae usually found at North America, Africa and Europe, but they are widely distributed across Asia. The fish of the family Cyprinidae, which is cyprinids, are commonly known as carps, minnow and barbs, and according to Nelson 2006, Cyprinidae is the largest freshwater fish family with more than 1,009 extant species in Southeast Asia. The family of Cyprinidae was divided into 10 subfamilies, which is Labeoninae, Probaninae, Taurinae, Smilogastrinae, Cyprininae, and many more. According to Young et al. 2010, subfamily Cyprininae is the largest subfamily with 110 genera. The genus Cyclocheilichthys first cited by Bleecker 1859. However, some species in this genus were described earlier and assigned under genus Puntius, such as Puntius enoplos, Puntius armatus, Puntius apogon, and Puntius ripason. Then, Cotelat 1999 investigated the origin of genus Cyclocheilichthys and found out that Bleecker 1859 was the one who first cited Cyclocheilichthys. According to Fishbase 2020, the fish genus Cyclocheilichthys only can be found in Asia, ranging from China to Southeast Asia. The 7 species of Cyclocheilichthys that distributed in Southeast Asia include Cyclocheilichthys apogon, Cyclocheilichthys armatus, Cyclocheilichthys heteronema, Cyclocheilichthys gentocare, Cyclocheilichthys lecleri, Cyclocheilichthys sinensis, and Cyclocheilichthys ripason. So, the target species in this study comprise 4 species of Cyclocheilichthys. The first one is Cyclocheilichthys apogon, or known as beardless barb, because of the absence of barbel in the mouthpart area. Zakaria Ismail 1990 stated that Cyclocheilichthys apogon body is compressed and relatively deep. The head is longer than the post-anal length. Ramboff 1996 also stated that Cyclocheilichthys apogon size is up to 15 cm and they has large dark spot at precauda. Cyclocheilichthys apogon is widely distributed in Southeast Asia, known from Indonesia to Burma, and it is found in almost in all major drainage in Peninsular Malaysia. They are usually found in various habitats from large lake to small pond, as well as small throughout large-sized river. Ramboff 1996 has stated that Cyclocheilichthys apogon typically found around surface such as plant leaf, branches, and tree roots where they search for small plankton and crustaceans. Second is Cyclocheilichthys armatus. Is known to have one pair of barbel. According to Zakaria Ismail 1990, all Cyclocheilichthys armatus specimens in Peninsular Malaysia has only one pair of simple maxillary barbel, but Sontirat 1976 stated that small number of specimens from Thailand also has a pair of minute roster barbel. Zakaria Ismail 1990 also stated Cyclocheilichthys armatus is widely distributed in Thailand, Laos, Cambodia, Vietnam, Sumatra, Borneo, and Java. They were commonly in Peninsular Malaysia, but not as abundant as Cyclocheilichthys apogon. It is usually found at midwater to bottom level in river and stream from Indonesia to Thailand and Cambodia. Their diet consists of zooplankton, small crustaceans, and other larval insects. Next is Cyclocheilichthys ripason. Cyclocheilichthys ripason is species that has 2 pair of barbel and a large black spot at the caudal fin base. Zakaria Ismail 1990 also stated that Cyclocheilichthys ripason body is compressed but not very deep, and the head is slightly longer than post-anal length. According to Zakaria Ismail 1990, Cyclocheilichthys ripason species is found in Thailand, Laos, Cambodia, Vietnam, Sumatra, Borneo, and Java. In Peninsular Malaysia, it was reported from Tembeling River in Pahang Drainage near Taman Negara. Cyclocheilichthys repassen is found at mid-water to bottom level in small river, canal, pond and reservoir from Indonesia to Thailand. Their diet consists primarily of insect with some aquatic macrophyte. And the last one is Cyclocheilichthys heteronema. Cyclocheilichthys heteronema is the species that has a single pair of maxillary barbel which is multiplied. This specimen is unique compared to other Peninsular Malaysia species of Cyclocheilichthys for having a shorter preanal length. The body is compressed but not very deep. The head is relatively small and its length much shorter than postanal length. According to Zakaria Ismail 1990, Cyclocheilichthys heteronema is found in Peninsular Malaysia, Thailand and Borneo. Cyclocheilichthys heteronema is found near in bottom large river from Thailand to Borneo. Next is my study site. So the fish of Cyclocheilichthys were caught in Peninsular Malaysia and has deposited to USM and UM. For this study, I went to UM and USM to collect data of the preserved specimen. For the my objective is to determine the unique morphological characteristic between species under genus Cyclocheilichthys by using the morphometric and meristic analysis. The material method is divided into two, which is lab work and statistical analysis. For lab work, I collect sample at USM and UM, and lab session were conducted to examine the morphometric measurement and meristic data. I observed 12 sample of Cyclocheilichthys apogon, 90 sample of Cyclocheilichthys armatus, 6 sample of Cyclocheilichthys repassen, and 10 sample of Cyclocheilichthys heteronema. And then I go for statistical analysis using SPSS software. I use one-way ANOVA to determine the significant difference of characteristic for each species and post hoc to examine how differ these four species is distinct from each other. And the result is I determine that these four species has their own special characteristic that differentiate them with another. The first one is Cyclocheilichthys apogon. Cyclocheilichthys apogon has absence of barbel. At the right one is Cyclocheilichthys armatus. As you can see, the Cyclocheilichthys armatus is species that have one pair of simple maxillary barbel. The next is Cyclocheilichthys repassen at the left. Cyclocheilichthys repassen species is the species that have two pair of barbel. And lastly, at the right one is Cyclocheilichthys heteronema. Cyclocheilichthys heteronema is the species that have multiplied single pair of maxillary barbel. Next is these two species, which is Cyclocheilichthys apogon and Cyclocheilichthys repassen. Both has dark blotch at the base of caudal fin, while Cyclocheilichthys armatus and Cyclocheilichthys heteronema has absence of dark blotch at the base of the caudal fin. And then this is the table result of measurement character between Cyclocheilichthys apogon, Cyclocheilichthys armatus, Cyclocheilichthys repassen, Cyclocheilichthys heteronema. The one that have p-value less than 0.05 show that there is significant difference between these four species. The characteristics that are significantly different between these species are total length, body depth, caudal peduncle length, caudal peduncle depth, dorsal fin height, and so on. And so the A, B, C in this table indicate that the difference between those species. For example, for total lengths, show that Cyclocheilichthys repassen, this one B, have obvious different length among among 4 species. This one A, B, and A. So this one have obvious different length. And then this is the meristic characteristic. Then the character that has significant p-value less than 0.05 between these species are lateral line scale, scale row above lateral line, scale row below lateral line, perpendicular scale row, predorsal scale, and total Guericke. For your information, perpendicular is number of scale row around caudal peduncle, starting with the scale with pore at the base of median hyporeal plate or at the end of vertebral at caudal area. As for discussion, Cyclocheilichthys apogon is lacking barbel and have large dark blotch at the caudal base and has fork-shaped caudal fin. Total of the total length is 58 to 170 mm and it have 16 circumpendicular scale row. Cyclocheilichthys amatus has maxillary barbel and the total length is 73 to 215 mm and have 16 circumpendicular scale. Cyclocheilichthys hypason has 2 pair of barbel and have 16 to 20 circumpendicular scale, and it seems to have oval dark blotch at caudal base and have 32 to 35 lateral line scale. And for Cyclocheilichthys heteronema, it have 1 pair branches, maxillary barbel, and lateral line scale of Cyclocheilichthys heteronema is 32 to 35. So some of the characteristics show there is significant difference within species, while some have no significant difference and a little has constant data within species. I assume that the 4 species exhibit different characteristics due to the different localities, type of preferred food, and abiotic and biotic factor. The conclusion is all 4 species have their own special characteristic that can make them spread in different species. So the purpose of my study have been achieved by looking at the species characteristic among these 4 species of Cyclocheilichthys. The limitation in this study, some of the characteristics among these species show no significant difference due to small differences and there are limited resources that focus on this species. So, in future, the future researcher can fill the gap of knowledge by using more precise statistical analysis to study the morphology of fish genus Cyclocheilichthys by its molecular analysis. So, for the question 1, why morphological characteristic determination of the species are important? For the first one is morphological characteristic determination of the species are important because the accurate identification of organism is important when measuring the biodiversity of an area. It is also a prerequisite when attempting to find out more about the ecology of a species. And without accurate identification, it is impossible to determine how many species exist in a given area. For the second question, the reason why the specimen need to be deposited in USM, and UMT, it is because there are museums at USM, and UMT that store fish specimen for the long time and can be used as reference for further research. For the last question is another reason why I choose to study this species apart from taxonomy is because there is limited previous study about this species and I would like to know more in detail about this species. The current status of this species is they were very abundant in Peninsular Malaysia and Cyclocheilichthys apogon is present at local food fisheries and in the ornamental fish trade. Thank you.